This is an ongoing discussion of exercise physiology. We are getting towards the end of Chapter 3 on bioenergetics. As we concluded the last part of this chapter discussion, we were talking about anaerobic mechanisms of producing energy in muscle cells, namely glycolysis and then fermentation. We said that if glycolysis occurs in isolation to produce ATP for muscle contraction, then fermentation is necessary, in which the pyruvic acid produced by glycolysis is converted to lactic acid in order to get the high energy electrons back off from NADH so that NAD can be restored and more cycles of glycolysis can occur. A more efficient process involves aerobic respiration, and we mentioned at the end of that section that in the mitochondria, the pyruvic acid can be oxidized the rest of the way to CO2 and water through a method that uses oxygen to produce a total of 34 ATP per glucose as opposed to 2 ATP per glucose for anaerobic glycolysis. So as the pyruvic acid is converted to acetyl-CoA, right? Pyruvic acid, we have two of them that came from each 6-carbon glucose, so we have two 3-carbon pyruvic acids, and there's an oxidation reaction just as this will enter the mitochondria, in which one more carbon is shed, producing CO2, and acetyl-CoA is the entry point. The 2 carbons, the acetyl group is 2 carbons, will then be added in the mitochondria to the first chemical of Krebs cycle, oxaloacetate. So that was a 2-carbon or a 4-carbon compound. We're adding those 2 carbons and we make citrate, which is a 6-carbon compound. That's not so critical. The main thing is we're delivering acetyl-CoA to the mitochondria, which will enter into a series of cyclic reactions, and in the process we're going to oxidize, get those last 2 carbons will be oxidized to CO2, and we're going to produce a couple of things. In that series of reactions, Krebs cycle, we're going to get an ATP out for each cycle, so 2 per glucose, because there's 2 pyruvic acids. But we're going to get out NADH and FADH, just like we did during glycolysis. These are the important products because the high energy electrons carried by NAD and FAD here are very valuable. They're very energetic. They have a lot of potential for making ATP. So Krebs cycle will give us a bunch of high energy electrons. And here's a representation of it. I don't want you to memorize this, of course, but it's good to see things sometimes, to visualize what's going on, and I'll just run you through what's going on here. Here's pyruvic acid or pyruvate coming from glycolysis up above. It's being oxidized to acetyl-CoA. You can see the CO2 being released. And acetyl-CoA in the mitochondria is added to oxaloacetate. That's that 4-carbon compound, and when you add 2 carbons, we wind up with citrate. A series of reactions ensues, restoring oxaloacetate again. So that's why it's called Krebs cycle. We keep on recapitulating oxaloacetate each time, which can then grab another acetyl group from acetyl-CoA and go around and around. In the process of oxidizing that citrate to form oxaloacetate, here you can see NADH being produced. We're capturing high energy electrons. You can see FADH being produced. We're capturing more high energy electrons. So that's really the main product of Krebs cycle. We do produce an ATP for every time around the cycle. It starts out as a GTP, which can then be converted directly to ATP. All right, so that's the Krebs cycle. We got a lot of high energy electrons. We'll actually take those the electrons from glycolysis also and shuttle them into the mitochondria. And what are we going to do with them all? Well, let's take a second first before we go there to mention what's this word fatty acid doing in here? Little arrow. And amino acids. Well, if you remember, fats are stored as triglycerides in adipose tissue and in muscle cells, and a triglyceride represents is made up of a glycerol backbone and 3 long-chain fatty acids. In order to utilize the fatty acids for energy, you need to break that thing back down. The fatty acids are released into the cytoplasm of the muscle cell or into the blood, and then through a process called beta oxidation, those long-chain fatty acids are are converted to acetyl-CoA 2 carbons at a time. So if you keep breaking off 2 carbon units, produce a whole raft of acetyl-CoAs for each fatty acid chain. The glycerol, incidentally, can be taken up by the liver and made into glucose. Gluconeogenesis, the production of glucose from non-carbohydrate sources. Protein is not really an important energy source for exercise, but proteins, excess amino acids, are deaminated in order to utilize them. The amine groups are gotten rid of in the kidneys in the form of urea, and we stick that right in the urine to get rid of it. And the remaining keto acid can enter into Krebs cycle, or it can be used to produce acetyl-CoA and energy. So excess amino acids can be broken down for energy. They're not really an important contribution during exercise. So this is just another little cartoon representation of that. When we get fatty acids released from adipose tissue, it's going to be in the form of free fatty acids in the blood or in the cytoplasm of the muscle cell. It'll be then entered into the mitochondria as an activated form, acyl-CoA. That just means the long chain is there. An acyl group is a long chain carbon chain. Right, so that's just all it's saying. We've got the long chain fatty acid be attached to this carrier molecule called CoA, coenzyme A. And then we go through a series of reactions called beta oxidation and trim off 2 carbons at a time in the form of acetyl-CoA. Okay, so that's right where we were talking about. We enter that acetyl-CoA right into the Krebs cycle, and that's what we're talking about right here. That arrow should really be pointing right to the acetyl-CoA. Boom, right into Krebs cycle. So that's where fatty acids enter in. They enter in at that level, at the level of the Krebs cycle. Here's a diagram which I'm not super in love with, but it does sort of bring together some of the conversions and pathways we've been talking about. The liver can store glycogen from glucose, polymerize it, and then later release it back out into the blood to be used by cells to produce energy. So this looks like it's all happening in limbo, but it's going to be happening inside a cell in your body. The cell's going to take up glucose, phosphorylate it, run it through glycolysis, right? When you take a glucose and produce pyruvic acid, we should have like maybe a circle around this indicating we're talking about glycolysis. And then finally, if we are able to do aerobic respiration, we'll take the pyruvic acid, convert it to acetyl-CoA, and bring it into the mitochondria and run it through Krebs cycle. And we haven't talked about what happens after that, but we will. Fat stores triglycerides. Those are adipocytes. Muscle cells also store triglycerides, and they can be broken down into glycerol and those 3 long-chain fatty acids. In the muscle cell, we know that the 3, if they're taken up by the muscle cell or coming from within the muscle cell, the fatty acids will be activated and then broken down to acetyl-CoA in the beta oxidation process, and then that acetyl-CoA can enter Krebs cycle. Here we talked about fermentation. If glycolysis occurs in isolation without using the Krebs cycle or the subsequent electron transport chain, we need to do fermentation, and so the pyruvate will be converted to lactic acid during high-intensity exercise, as we'll talk again about later. So later, once you get tired, right, in the presence of a lot of lactic acid, you're going to be fatigued, you're going to stop exercising and take a break. And it's during that break that the lactic acid will be converted back to pyruvic acid. That's what this arrow is for here. You're not going to leave that lactic acid in the body as soon as during exercise, and then during the resting period, you will continue to get rid of that lactic acid and convert it back to pyruvic acid, run it through the Krebs cycle, and get all the energy out of it. Yes, during high-intensity exercise, amino acids, proteins within muscles and other tissues can be broken down, releasing the amino acids, which can be the amine groups are gotten rid of in the urine in the form of urea, and then the keto acid that's left can be converted to acetyl-CoA and used for energy. But again, that's not really a major thing. We hope that's not a major thing. We don't want to break down all the protein in our muscles during exercise. All right, so Krebs cycle gives us all these high energy electrons. Now we can actually utilize them. Oxidative phosphorylation is the process by which all those high energy electrons are going to be utilized to make ATP. from both glycolysis and from Krebs cycle. It has two steps. The electron transport chain is going to set up some potential energy, and then we're going to use that energy to drive the actual ATP production. So the high energy electrons are taken from NADH and FADH and passed over to some proteins, some cytochromes are called, in the inner membrane of the mitochondria. The mitochondria is like a double-walled thing. It's got an outer phospholipid bilayer, like most organelles do, but then it's got another inner one. So it's double layers. And all these carriers of electrons are in the inner mitochondrial layer. And the electrons are going to be handed from cytochrome to cytochrome in a chain reaction. So redox reactions we talked about, right? We're going to keep on oxidizing some molecules and reducing others and passing those electrons from NADH and FADH down a chain of cytochromes. Every time the electrons are handed down, some energy is released, and that energy is going to be harnessed by those cytochromes, by some pumps that are in the membrane. So we're going to take the released energy and pump hydrogen ions across the membrane. Remember we talked about the sodium potassium ATPase does something like that. We called it pumping, moving ions from low concentration to high concentration. In this case, we're not using ATP to power that active transport process. We're using the energy from the high energy electrons, but it's very similar. So anyway, we're pumping hydrogen ions from low concentration inside the mitochondria to high concentration between the two membranes. So it creates a big concentration gradient across the membrane. Let's take a look. I'm going to show you this diagram first before we go back to that picture. So that statement. So here we are, this innocuous looking tiny little typed NADH. We're taking the NADH, which has grabbed those high energy electrons, and it's going to pass them off to this cytochrome here, this pump, and the energy that's yielded from that electron is going to be used to pump hydrogen ions across the membrane. Here's the inner membrane right here. This is the outer membrane of the mitochondria. So we're pumping hydrogen ions across. Then the electron is handed down to another, through another intermediate, and then to another pump, and more energy is released from it, and another hydrogen ion is pumped across the membrane. And then we take the same electron, which is now a little lower in energy, and hand it off a couple of times to a third pump, and it gives off yet some more energy. Now it's getting down to some pretty low energy. But every one of these pumps hydrogen ions across the inner membrane, forming a concentration gradient. In other words, a concentration difference. Look at all the hydrogen ions in here compared to out here. Also, we're getting the electrons from FADH here. You can see they just entered a different point. All right, so we got this huge hydrogen ion gradient, and as the hydrogen ions push their way back through the membrane, diffuse back across the inner membrane to the inside of the mitochondria, they drive an enzyme called ATP synthase, and it just physically makes ATP by taking ADP and forcing that inorganic phosphate onto it for that third phosphate bond, that high energy bond. And the equivalent is that for every NADH, every pair of electrons from NADH, we get 2.5 ATPs from it. And for every FADH's pair of electrons, we get 1.5 ATPs from it, right? Because the FADs are coming in a little later. 2 pumps are going to be activated by FAD electrons. 3 pumps are activated by NAD electrons. Now, the reason we have this funky thing of partial ATPs is because it's not a direct thing. Chemiosmosis is this process by which you form this gigundous hydrogen ion gradient, and they are going to diffuse, or think of more of them forcing their way through a revolving door. And as they force their way through this machine, this ATP synthase enzyme, it actually turns and mechanically, mechanochemically does this reaction. It takes ADP and forces that third phosphate onto there, forming ATP. Now the hydrogen ion has diffused across into the inner membrane, and it will be pumped right back out again, as long as we're continuing to do this process. So that's chemiosmosis, is this movement of the hydrogen ion back through the membrane, driving ATP synthesis. This is the electron transport chain. This is chemiosmosis happening right here. So those things together represent oxidative phosphorylation. It's got those two parts. Electron transport chain, taking the energy from the electrons to pump hydrogen ions, and then chemiosmosis, the driving of the ATP synthase. Wonderfully magical stuff happening here in the mitochondria. Okay, so we don't have a quick balance sheet right here. Eventually we'll come up with a balance sheet, but just to remind you, ultimately we're going to wind up with 32 to 34 ATPs for every glucose molecule, including the ATPs we got from glycolysis 2, the NADH from glycolysis, electrons being introduced here, and the electrons being introduced from Krebs cycle, as well as those ATPs from Krebs cycle. Add them all together and you wind up with 32 to 34 ATPs for glucose. Pretty awesome. Free radicals are highly reactive chemicals or molecules that can be formed in the cell through a number of processes, one of which is the electron transport system. Occasionally electrons that are intended to go down this chain get diverted off and they react with oxygens to produce some very nasty, poisonous, reactive oxygen species that are called. And there's a number of different forms, but also free radical. That's just one particular type of chemical form. Whoops. Got fired up and pressed my mouse. So free radicals, the problem is they're so reactive, they just react with stuff they come in contact with. Lipids, proteins, DNA, and they make damage. They change the chemical properties of those species. That's not good. So in order to prevent them from being poisoned all the time, cells maintain a pool of antioxidant molecules so that when reactive oxygen species form, they can quickly be gotten rid of, ameliorated by antioxidants. That's why you hear all about antioxidants today. Let's take some antioxidants as a supplement because truly it can help get rid of reactive oxygen species that are formed inadvertently or during the immune system function in the body. Exercise promotes the production of free radicals within muscle cells. Now, we've just said that's not so great, but it turns out that we just remember before when we talked about hormesis, we said that some kind of deleterious stuff happens when we challenge the body with various things, including exercise and muscle contraction. But at just the right amount, those deleterious things that happen produce good reactions, good adaptations. And so reactive oxygen species production actually triggers some cell signaling pathways in muscle cells that lead to adaptation and growth of the muscle cells and increase in performance capabilities of those cells. So just to bring that into focus, and we'll talk more about that when we talk about muscle cells themselves. We'll do a section on adaptation. So here's our balance sheet real quick. I probably should have moved that up a bit. That's okay. So glycolysis produces 2 ATPs plus 2 NADHs, and we saw that that's going to produce 2.5 ATPs for NADH. And so we add those together, and we get some ATPs being added up. We get some energy out when pyruvate is converted to acetyl-CoA. We get NADH from that. The Krebs cycle, the authors love to call it the citric acid cycle. I prefer to call it Krebs cycle in honor of Hans Krebs. So anyway, we produce, yes, we produce ATP directly, but we all have 2 for every glucose, 1 for every cycle. And then we produce a bunch of NADH and FADHs. Those are the high energy electrons. And then by the time we pass those through the electron transport chain and chemiosmosis happens, we wind up with 32 ATPs per glucose. Why, oh why do we say 32? I keep telling you 34, don't I? Let's go back to a slide that I edited since you saw part 2 of our discussion. I tidied up this slide a little bit to show you something that I didn't really address before, and it's not critical, but it's interesting nonetheless. If you want to store glucose molecules in the form of glycogen, you have to invest in ATP to do it. So here's glucose being brought into a muscle cell, say. It has to be phosphorylated first using an ATP before we can run it through glycolysis. And then as it runs through glycolysis, we have to invest another ATP. Remember we talked about the investment steps of glycolysis. That's what it is. 1 ATP, 2 ATPs. And then finally we get 4 out for a net 2 ATPs. Well, if you decide to store the glucose first for a while in the cell as glycogen, so that when you have your race or whatever, your exercise bout, it's there for you, you have to invest yet another ATP to do that. So we still have the 2 ATPs we're going to have to invest to do glycolysis, plus a third one. So you can think of it as, if you store glucose as glycolysis, you're going to have to pay an extra ATP per glucose, and we're going to subtract off from the total then more ATP. So ultimately, with everything considered and everything we know about the biochemistry of bioenergetics in cells, there's a net 32 ATPs for every glucose, considering it's coming from glycogen. Okay. What about the efficiency? How fun is that? We talked about the efficiency of exercise. We said that if we take the external work done and divide that by the total amount of energy the body had to consume in the process of doing that exercise, we can figure out the efficiency. We can do the same thing with respect to cellular ATP production. If we make an ATP from glucose, say, we know how many ATPs we get, and we know how much energy is available in each ATP if we break off that third phosphate to utilize the ATP, 7.3 kilocalories for every mole of that ATP. And then we take some glucose and burn it. Burn it in the bomb calorimeter, an instrument that can measure all the heat released as we go from glucose right to CO2 and water. Every bit of the energy is going to be measured then. That's the total glucose content. We'll find out that, our process of forming AT is around 34% efficient. Incredible. What happened to the other 2/3 of the energy that was originally stored in the glucose molecule? Released as heat. So it's a good thing to keep in mind. I'll probably ask you that about a million times. How much of the energy from your fuel molecules is lost as heat in the production of ATP? 2/3, 66%. I'll throw in one more thing. Eventually, we're going to take those ATPs and use them for muscle contraction, among other things. When that happens, when we hydrolyze the ATP back to ADP, 2/3 of that energy is lost as heat. So a lot of heat being produced in muscle cells during muscle contraction. That's the challenge of our body, to release that heat from the body so our body temperature doesn't go through the roof as we exercise. Okay, this is great. We now know a lot about how muscle cells produce energy. How do they produce energy from glucose molecules? How do they produce energy in anaerobic conditions and aerobic conditions? How do they produce energy from fatty acids, beta oxidation and acetyl-CoA being run through the Krebs cycle? Now we have an important question. How do they know when it's time to do all this stuff? What are the controls? What are the signals that tell the cell now's the time to start undergoing glycolysis and Krebs cycle and electron transport? Well, it turns out that many metabolic processes in cells are regulated by the products of various chemical reactions. So when we make a lot of ATP in cells, we're going to find out in a minute that that's going to inhibit the processes by which ATP is made. It's going to inhibit the function of enzymes, so-called rate-limiting enzymes, because if we already have a rising ATP level in the cytoplasm, no need to keep on going. That's what shuts off the production, so that when you're sitting in the infield of the race, huffing and puffing, eventually you feel good and you stop huffing and puffing. You restore all that ATP that was originally in the cell. You recycle all the lactic acid that had been produced back to pyruvate and make ATP, and everything's cool, and so then you stop and you go back to just resting conditions. That's because ATP is going to inhibit the rate-limiting enzymes in glycolysis and Krebs cycle. Then we start exercising, and we start hydrolyzing ATP to produce ADP and inorganic phosphate. Those, not only does our ATP go down and stop inhibiting those rate-limiting enzymes, ADP and inorganic phosphate tend to activate those rate-limiting enzymes. They're the slowest ones in a series, right? And so this is what turns on all these productions. You use ATP, boom, the machinery just starts up. And here are the rate-limiting enzymes in some of the systems. You don't need to memorize those enzymes, but it's just interesting that, let's say glycolysis. What's the so-called rate-limiting enzyme, the slowest enzyme, the most difficult enzyme reaction to proceed? And what turns it on and what turns it off? When we get a lot of ATP in the cell, shuts off glycolysis, shuts off this enzyme. A lot of creatine phosphate, we're done. Shuts off this enzyme, right? What turns it on? When ATP goes down and creatine phosphate go down, but in addition, when adenosine diphosphate begins to accumulate, right, because we're burning through ATPs, that turns on phosphofructokinase, and glycolysis is underway. All the rest of the enzymes just work. Whenever this one starts, then all the rest just keep doing their jobs, and we produce the products, pyruvate and high energy electrons. About Krebs cycle, same exact thing. Rate-limiting enzyme is isocitrate dehydrogenase, and you can go back and look at the light blue map of Krebs cycle and see right where that sits. And what turns it off? ATP. When ATP levels start to rise in the cell, we don't need to make more. Stops Krebs cycle. When we start burning through ATP as we exercise, ADP begins to accumulate, ATP is going away. Those two things both activate isocitrate dehydrogenase, and again, all the rest of the enzymes just, when their substrates appear, they just catalyze the reactions. But this first one here can be shut off in order to control the Krebs cycle. We don't run it when we don't need it. And finally, electron transport, same exact thing. Cytochrome oxidase in the electron transport chain is turned off by ATP. When we're all done making ATP, we got plenty. The cell has its standard amount in the cytoplasm that it likes to keep on hand. And then when we start exercising, we burn through that and we produce ADP, turns on electron transport. So don't mean to beat that to death, but it's cool to know that it's not just random. As soon as we start taking away ATP as we exercise, all the machinery starts to whir into motion, and we start making electrons, or start making ATP by all the methods we've just learned about. Here's a generic picture of that happening. Most metabolic processes in your cells, there's a product produced at the end of, say, a series of chemical reactions, and in our case, we've just been talking about ATP. It feeds back and inhibits the rate-limiting enzyme of the series. So whatever it might be, you can keep this in mind for virtually any product that's produced in your cells by a series of chemical reactions. And if that product is available in large supply, you don't need to keep making more. And its very presence will bind to the enzyme in its own pathway and stop the enzyme. Once we start using up this product and it starts to go down in the cell, then enzyme A, the rate-limiting enzyme, is no longer inhibited, and it starts back up again. The machines start whirring and we produce the product. So that's just the way that product inhibition acts as a feedback inhibition mechanism to control the concentration of many substances in cells. This is a thing that's become popular, apparently, an analogy of how these energy-producing processes are regulated using this idea of gravity and hydraulic pressure, right? These cylinders are supposed to represent vats of water acted upon by gravity, and the push of, as we use up some products from these little tanks, then we're going to open up some valves and let the water from this fuel source push its way through high energy here to replenish the energy of these tanks. Actually, this isn't the greatest example of this. There's some much better examples. I was going to put one in here from a research article. They thought you'd be overwhelmed if they drew in like almost like a float inserted into this tank attached to a valve, so that the opening up of this valve actually occurs as this tank goes down. A float attached to a lever opens up this valve and starts letting some of the fuel molecules be oxidized to produce energy, and likewise this tank has a float in it, and so forth. I'm not in love with this thing. I think you can understand the idea that the rate-limiting enzyme and the processes we've talked about, glycolysis, 10 chemical reactions in the cytoplasm, Krebs cycle, a series of reactions in the mitochondria, and electron transport system, you can appreciate that we can stop that thing once we produce a lot of ATP by inhibiting enzymes, and we can activate that whole thing once we start to burn through the ATP during exercise, and the ADP levels start to rise. That's what we get when we hydrolyze ATP for muscle contraction. So if you love this thing, just gaze at it and enjoy it. If you want, you can look up the hydraulic model of oxidative phosphorylation, and you can find some research articles that will have cartoon illustrations of the same thing, only probably more elaborate. So if you like that, that's fine. Last thing to talk about in this chapter, we now know that we can do anaerobic glycolysis in isolation and fermentation to produce a little bit of ATP at a very rapid pace, though. Millions of cycles of glycolysis, but we'll do fermentation and lactic acid will build up. Or we can slow things down and run the pyruvic acid through the Krebs cycle and so forth and produce a lot of ATP by aerobic respiration. So when does this happen? How do we know? How do we choose between those things? Well, the choice comes from the type of muscle cells, essentially, that are going to be involved in the processes. This slide says in words what this slide so elegantly illustrates in these two tower-like bar graphs. When you're doing the highest intensity exercise, in other words, moving your muscles the fastest they can go, the fastest, say, swing of the baseball bat you can possibly muster, and maybe even a little bit just below that, some fast sprinting, as fast as you can sprint, that's going to be accomplished by anaerobic energy production entirely. It turns out that the muscle cells that you call upon for the strongest, most vigorous output from your muscles are a type of muscle cell that's just what they do. They do anaerobic glycolysis and fermentation. And it makes total sense. What's going to happen? We're going to produce a lot of lactic acid. How long can you sprint your very fastest? Not very long. After about a minute at the very, very most, you're all done with your high anaerobic respiration source of ATP. You're going to sit on your butt and rest, and what's going to happen? We're going to recycle all of that lactic acid back to pyruvic acid, etc., like we talked about, and you're going to restore the ATP levels in your cells, and everything's going to go back to normal. If you think of the opposite extreme and do something extremely submaximal, is a word we like to use a lot, some very low intensity exercise, walking, think. They put a marathon run. Well, you could do some walking and do just aerobic respiration. If you're in great shape, yeah, you could run a marathon at a certain speed and use just aerobic respiration, but most of us aren't going to do that the first day out. All right, anyway, point is, low intensity exercise uses aerobic respiration entirely, theoretically, and not anaerobic respiration. But in most exercise situations, you're doing some of each all the time. Let's look at something here. 800-meter race. You're using about 50 to 60% anaerobic glycolysis and about 50% aerobic respiration. You're still producing some lactic acid. You bet you're not going to go as fast as you can in your fastest 800 dash any longer than that. That's as far as you can go. You can run about 2 minutes at that rate. You're getting some benefit of the high efficiency of aerobic respiration, but you're also producing a bunch of lactic acid. So the higher the intensity exercise, the greater the fraction of ATP that's produced by anaerobic respiration and fermentation. And as you go to lower and lower intensity exercise, the greater and greater fraction or percentage of ATP that's being produced by aerobic respiration using the mitochondria that we can do theoretically all day long. There's no actual limit to how long you can continue to do aerobic respiration producing ATP from your fuel molecules in the mitochondria. There are some things that happen that ultimately will produce muscle fatigue, but you can adapt to training of aerobic forms of exercise, and ultimately once you reach a certain level of fitness, you can just run forever. As long as you can shovel in food and stop and eliminate waste from your body and keep yourself hydrated, you can run forever. I think the record, as I last heard about it, was for trail running was something like 340 miles, just to beat the world record and not go on any longer. It's a pretty grueling experience, but somebody showed that you can run for days at a time without stopping using aerobic respiration. All right, so that's our story. What a wonderful chapter it is. It brings to light so many things that happen during exercise, and it'll be a foundation for pretty much everything we talk about when we talk about muscles and how they work and why we have muscle fatigue and so forth and what happens during adaptation. It's going to be great. So join me for the next chapter.